Do you want to crack NEET in 2024? And if yes, so if you want to crack NEET 2024, you have to get excel in lots and lots of topics. And the topic which has maximum weightage today, we are going to start that topic, which is Mendelian genetics. So hi everyone, this is Bassi, your biology educator, and today we will be going to start with a very important chapter, which is genetics. Already we have discussed about some basic concepts, about some basic terminologies about this particular chapter. So today we will be moving forward and we will be starting with the Mendelian genetics, right? So if you have not watched the basic videos, like what are the terminologies like alleles, what do you understand by genes, if you are not if you have not watched the video, go back, watch that video after this class. Okay, so while we are starting here about the Mendelian genetics, we should know about the person, that who Mendel actually is, right? Mendel was a very great scientist, and we will be knowing his work in this chapter. So the very first thing, let's discuss about the Mendel, like when he was born. Mendel was born on 1822, the year 1822. You just remember year, and he died around 1884, right? So what was the birth year? 1822. And at what year the Mendel died? Mendel died around 1884. Now, this person, as we discussed in the previous class, this person is considered to be as the father of genetics. Now, why we call him as a father of genetics, we are going to discuss in this particular session. Before starting, Mendel was really a very great scientist, and he had lots and lots of experiments, but his work was not recognized. The work was recognized after his death. So sometimes we get demotivated, right? Like, oh my god, I am preparing for NEET, I am giving mock test and all, but still I am not getting the results. So that doesn't mean I am telling you that you'll get your results after your death. No, not like that. But what I am saying is that you should keep working. You should never stop. If you have that zeal, if you have that consistency, one day the fruit will come towards you. Okay, let's get back to the topic. So this person, which was Mendel, he is known as the father of genetics, and his experimental organism was the Pisum sativum, and since it's not underlined here, so I will underline this Pisum sativum. The common name for this Pisum sativum is garden pea. Yes, the pea which we actually see, that is garden pea. Now, he completed his experiment around 1863, and he started his experiment 7 years back. Like, he took 7 years to complete his experiment. So, what should we do to find the date on which he started the experiment? So, let's write here 1863 minus 7. Now, apply your brain, apply your mathematics brain. So 1863 minus 7 will be what? Anyone. It will be 54, 55, 56, 57, 58, 59, 60, 61. No, then what? It will be around what? It will be around, just a second. Is my voice audible to everyone? Let me check, guys. Okay, my screen is not visible to you. That's the issue. Great. 1 second. I will clear this thing out. Just a second, everyone. Just a second. If it's not visible, I will reshare. I'll definitely reshare. Okay. I think so. There is some issue, and nope. Still, it's not visible. Okay. No issues, no issues. Just a second. We are going to see this. What's actually happening? Why my screen is not visible? Just a second, people. If you're not able to view my full screen, then I can make this screen full, right? Yeah, I am pretty intelligent. Okay, no issues. Just a second. It's a little technical problem here. Okay, now I think so. You will be able to view my screen no matter what. So just let's check. I'll restart. Don't worry. If you have not understood anything, I'll restart. Yeah, now my screen is visible to you all. That's very good. You can see my screen. Okay, let's start again. So in this session, we will be going to discuss about the Mendel, right? And if I am talking about the Mendel was what? Mendel was a great scientist, and he was born on 1822. The year of birth is 1822, and he died in the year 1884. Now, he did lots and lots of work, lots and lots of experiments on pea plant, and that's why we call him as a father of genetics. Now, what's the basic reason calling him father of genetics was that he gave so many terminologies, he gave so many things which no one knew about genetics. He literally experimented and created a field which is genetics. So yes, that's why this person is called as the father of genetics. Now let's move on and let's talk about his experimental organism. So his experimental organism was Pisum sativum, which is garden pea, right? Pisum sativum. Now, you will be amazed to know that he completed his experiments in 1863. The experiment was completed by the Mendel on 1863, in the year 1863. But unfortunately, he was not able to get recognition. His work was not recognized by anyone. So, but in 1900, some scientists came, they restudied the Mendel work, and they saw that, wow, the thing which Mendel has done, it's epic. No one would have done that. So they published their work in 1900. Clear? So we discussed about Mendel. Now let's move on towards the next thing, and let's talk about the success of Mendel. Like why Mendel is so successful, why his experiment was so successful. There might be some good thing, that's why his experiment is now recognized as worldwide. So let's write here what was the need or why the Mendel is successful. So see here, Mendel success, right? So if I talk about the success, very first thing is that what he applied, for the very first time he applied mathematics, analytics and logical things in the biology, right? So let's write here, he applied maths, reasoning, analytics, statistics, all these things in biology. Now, second was what? Like his progeny, like whatever the experiment he has done, just forget about the word which I used, progeny. We'll discuss it later. Whatever the work he has done, he had large number of plants, right? So can I write here large size? The experiment was of large size. Now you might be thinking that, sir, if it's fine, if it's large size, we can totally understand. But how this can be the reason for the successful of Mendelism? See, let's suppose that I have to do an experiment. Let's suppose I go and I talk with two of the students, two dropper students, that, hi, how did you crack NEET during the drop years? So both of them, let's suppose they were not able to crack NEET. So my experimental size is very low, right? And according to these two students, what I will say, that my result will say that a dropper can't crack NEET, right? But if suppose I went to a broader audience, like I took 7,000 students, I took 10,000 students, or I took lakhs of students, then I can get a proper percentage. Then I can give you the exact results. So this thing is what? This thing is, large size is basically ensuring that the experiment is error free. So his experiment was what? His experiment was error free, and that was the main reason for the success of Mendel. Now see, next, he didn't said like, okay, he just did things and he was not able to give the proof, nothing like that. He gave the theory and he proved it, he tested it. So all these things supported Mendel's experiment. So he gave theory also, and if someone needed the proof, he proved it also, and he tested those experiments. So all these things combining make Mendel a successful scientist. Now, next, if I'll go, then what I should say here is that why Mendel was not recognized before 1900, right? Sir, you are saying that the Mendel was so successful. Yes, we know. But why the work of Mendel was not recognized before 1900? This can be a case, right? See, what was the conditions why the Mendel work was not recognized? So let's quickly see that also. There were some reasons why the Mendel work was not recognized, and you can also just imagine like why, what could be the reason. Number one thing was, let's see, there were no communicating media like today, right? Let's suppose I want to say anything. What I'll do, I'll just go on YouTube and I'll just go on Instagram, I'll post things. But during that time, there was no social media. So there were no communication between anyone. So Mendel did what? Mendel just published the things, and no one could have connected with them. No one could have asked Mendel that what you have done, right? So there was a lots and lots of communication gap between during that time. And this is one of the reason why Mendel work was not recognized during that time, right? And next, what was, there were some complex terminologies like factor, right? He gave the word factor, and right now we know that this factor is known as gene, but during that time it would be very difficult to make someone understand that what actually factor is. Just imagine, let's go to that 1880s eras and we will be talking about this thing, that a person standing in front of a camera and you will be able to see him, you will be able to hear him from your home. So this will be like very, very like unrealistic to them. So the new introduction of new terminologies by the Mendel made the audience very confused. They were not able to understand, and there was a valid reason that Mendel was not able to show the factors. Like if suppose that I'll say that I have a flying car, the very first question you will ask me about, sir, show me the flying car. If suppose I am able to show you that flying car, you will believe that okay, flying car exists. But the same question was asked by the Mendel, that where are the factors, and Mendel was silent. Mendel couldn't show the physical existence of factor. So I can write here physical existence of factor was not there. Or I should say physical existence of factor was not shown, right? Why? Because of microscopes and all, there were many issues during that time. The world was not very much advanced. So this was the reason why Mendel's work was not recognized, right? And after that, what happened during this thing? During 1900, what happened in 1900 that Mendel got the recognition? You might be having this question also, right? Like, okay, fine, 1884 tak Mendel was not able to publish his work very in a worldwide, but what happened in 1900 that he got recognition? So during 1900 there was basically three scientists, okay? 1900 there were basically three scientists who were doing the same thing. Like, they were trying to gain knowledge about the genetics, they were trying to do experiments regarding the genetics, and they read the Mendel's theory. So there were three scientists. One was Tischermark, and let's write here. The other was Hugo de Virus. And the third one, we had what? We had Karl Cornut, okay? These were the three scientists who recognized Mendel's work, and all of them published his thesis in 1901 in a book called Flora. So all these scientists recognized Mendel's work and they published it in 1901. And this is the time when Mendel got this tag, which is father of genetics. So although Mendel was not successful when he was alive, but without the Mendel, this genetics would not be possible. So let's salute to this person, having so many failures also, still he managed to do these things. It was a very great job at that time. Now, let's move on and let's see like why Mendel chose Pisum sativum only. Like, he did the experiments, right? Now we have to study about the experiments also, and let's see that why Mendel chose Pisum sativum. Why not any other organism? Why not an animal? Why not a human being? Why not me? Why not you? Why? So Pisum sativum was what? Pisum sativum is a plant basically, right? So doing reproduction in plant was very easy. Just we have to bring pollen grains, anther, and if we are able to place the pollen grain into the stigma, we can have that pollination and reproduction can be easily done. But for an animal, it is a very complex process. You have to arrange sperm, you have to take out the eggs and do the fertilization. So that was a very complex process during that time, and it is complex process for now also. So that's why number one reason he chose a plant. Now, why Pisum sativum? The very first thing, they can be easily grown, okay, like anywhere they can be grown. Now, second most important point is they were having contrasting traits. Now, what does this contrasting traits means? I'll show you this with an example. Let's suppose that we have something like, do I have something? Yes, I may have something. See, this is a white pen, okay? This is a complete white pen. So if I talk about, if I keep this thing here, you can see that, okay, from here, black thing is there, and from here, the white thing is there. So you are able to distinguish this black color and this white color, right? Yes. So this means contrast. And if suppose that I'm wearing a black t-shirt and my background is also black, right? So it's not very easily to distinguish. Clear? So that's what I meant by saying the contrasting traits. So Mendel was like this pea plant is having a like two. I'm just going to give an example. Let's suppose one of the trait was tall. So the plant is tall and the other was short. There were no medium things, right? So it was very easy to check that, okay, this one is tall and this one is short. So that characters were easily distinguishable, and that's what we mean about the contrasting traits. Now, let's see shorter lifespan. So what does this mean? How this will going to help us? You can say that, sir, if it's shorter lifespan, then maybe it will create a problem to Mendel. But no, shorter lifespan do not create problem to Mendel. Why? Because if we are going to have shorter lifespan, then more progeny in shorter duration, right? Yes. So more and more number of generations, more and more number of progeny, more and more number of results. So shorter lifespan was also a important character why he chose Pisum sativum. Next, obviously large progeny. If we have shorter lifespan, progeny will be large, and in one reproductive cycle also, this plant was able to produce lots and lots of seeds, and each seeds will give rise to the plant, right? So we had large size progeny, but it was not possible in some animals. Let's take a human being only. We know the gestation period of human being, right? So let's suppose Mendel thought, okay, leave Pisum sativum. I will be using human as my experiments. Although we can't use humans, okay, it's not acceptable by laws. But let's suppose he would have done that, right? So first of all, he has to wait for that human to become sexually mature, which will again take 16 years, 17 years, 18 years, right? And when the human is sexually matured, then what? Then 9 months again, then the baby will come. And to study the next generation, he has to again wait. So till then, Mendel's life cycle will be over and he would have died. So that's why he has not chosen those animals or those plants whose life cycle was very, very large, very high. Why? Because the experiment would have costed lots and lots of time, and which would be practically impossible. Next thing is that naturally self-pollinated. Now, what do you mean by naturally self-pollinated? Naturally self-pollinated is what? Like the pea plant is having anther and stigma on the same flower, right? So they can do the self-pollination, which was naturally possible. And what was the benefit for this self-pollination? See, if suppose that one plant is tall, right? And that plant is doing continuously self-pollination. Let's suppose this is anther and this is stigma. So anther, from the anther, pollen grain is released to the own stigma, which is known as self-pollination, and this thing is continuously done. So what will happen? This plant will produce the progeny just tall, and that's how we can get a homozygous dominant. Homozygous dominant. Now, what is this homozygous dominant? Which means both the alleles will be same and they will be dominant. So this thing was helpful in creating a homozygous, or I should say a pure line, right? Or I should say a pure line. Now, let's move on and let us say that this plant can also artificially be cross pollinated. Why? Why do we need cross pollination? Let's suppose I want to study the interaction between a tall plant and a short plant, and for this we need a cross pollination. And how Mendel achieved it? By two methods he achieved. One was emasculation and the second was bagging. Two methods was done by Mendel. One was emasculation and the second was what? Second was the bagging. So what he has done during the emasculation and bagging, what and how? See, very first thing, if I talk about the structure of a flower, so we have a, what is this? Come on, let me know in the comment section fast. If I talk about this structure which you are seeing here, what is this? This structure is particularly the female reproductive part, right? Which we call it as a carpel. Now, After this, we have what? We have the anther, and then we have the beautiful petals here. So let me draw the petals as well. And here I have, this is the petals. Okay, my drawing is not good. It's good. Don't make fun of my drawing. I'll redraw again. 1 second. So let's have the petals like this. And then after the petals, what do we have? Sepals, right? So these are the structure. And in this structure, we have what? We have two essential parts, which is the reproductive system, which is male and female reproductive system, right? So to avoid self-pollination in the plants which he was about to do the cross-pollination, what he has done is that he just cut this anther. He just cut the anther. Now the flower is left only and only with the female part. And removing of anther is what is known as emasculation. The name itself suggests, right? Masculine, emasculine, removing the male characters, emasculation. So we can write here removal of, 1 second. Yes. Removal of anther. Now, when you are gonna remove the anther? After maturation? When the anther is matured and now anther is able to produce the pollen grains and can do the pollination? No. Why after the maturation? Before the maturation you have to remove, then only it will work. Now, if you are going to remove it after the maturation, then what's the need? After the maturation, the anther is matured, pollen grains has been released, pollination and has been done, and then you are going and removing the anther and you are saying, oh my god, I have done the emasculation. But that will not give you any kind of benefits. So when do you have to remove anther? Before its maturation. So removal of anther before maturation is called the emasculation, right? Emasculation is this only. Okay, now what does it do? What does it do? Sir, it prevents self-pollination. It prevents self-pollination. Yes or no? It prevents self-pollination. Clear? Now, so we have removed the anther. Okay, that's fine. But what if we remove the carpel or the female reproductive part? So basically we don't remove carpel. What we do is that, let's suppose that this is the female reproductive part, which is carpel, and it has 3 parts, right? If you remember, this part was known as stigma. Yes or no? This part is known as stigma, and this part is known as style, and this part is known as what? Ovary. And inside the ovary we have ovule and all those things which we are going to discuss in the sexual reproduction in flowering plants. Yes. So what we do is that we just take a plastic like structure and we just cover this stigma. Done. This is known as what? This is known as bagging. So stigma is basically what? In bagging we are not removing the stigma, but what we are doing, stigma is being covered. So stigma is covered by using, so can we use plastic here? No, actually we are not using the plastics, we are using butter paper. By using butter paper. By using what? Butter paper, right? Why we are doing this? Why we are doing this? Anyone in the comment section? Why we are doing this? Just to enjoy? Just to play? No. We are doing this so that the pollen grains, if the pollen grains will come also, it will just stay here. It will not be able to go inside and pollinate, right? So what does this bagging do? It prevents the pollination. Or I can also write that it prevents the deposition of pollen grains into the stigma. But you can understand pollination. What is pollination? Transfer of anther. Transfer of pollen grains from anther to the stigma of the plant, right? So if I have written pollination, that means pollen grains cannot be what? Cannot be come and land and deposit on the stigma. Why? Because we have created a barrier with the help of butter paper, and that is known as bagging. So Mendel had this kind of ideas, right? Mendel was very intelligent, not like us. He was a very intelligent person. And now so we are like, okay, do we have to do some experiments? No, sir. We will just copy and paste from everywhere, right? This is the thing which we are doing. But yes, if you will become a doctor, I would like each and every one of my students to at least give some time for research and thing, because your single research can change the whole thing. So don't underestimate the power of research, right? Now, let's say that again, we discussed why he chose the Pisum sativum, right? And we got to know that there were some large progeny, there were some contrasting characters, blah blah and blah, and everything we have read, right? But we will be focusing on those 7 characters, contrasting characters, and these 7 contrasting characters you have to learn it. It's not just for I have not written this just to view. This should be learned, right? And many of you will have confusion that which is dominant, which is recessive, flower color, whether violet is dominant, whether white is dominant. Oh my god, so confusing, but I have got you a trick, so don't worry. Okay, now here, if I talk about, let's make a table. So first of all, let's have flower. Okay, let's have flower color. Okay, or I can make the table here only. Just a second. Okay, we have the, let's try the dominant and recessive thing here. Okay, now if I talk about this flower color, so in the flower color, which one is dominant? Dominant one is the purple, dominant one is the purple, and recessive is white. Okay, now let's go with the seed color. So if I talk about the seed color, so what is here? Seed color dominant is yellow and green is recessive. Next, pod plant height. So plant height, we all know, plant height will be tall, dominant, and recessive will be short. Now next we have pod shape or fruit shape. What is that pod or fruit? The outer covering of the pea is basically the pod or fruit. Okay, so pod is what? It can be inflated. It can be inflated like this, or it can be constricted. You might have seen some peas from which you can from outer only you can see how many peas are present, right? So it's just constricted, and inflated is like kind of some air kind of thing is present and it's fluffy, and constricted is what? It is very close. It seems like it is just attached to the peas, right? So this will be constricted. Now next is flower position. Flower position is basically what? Flower position is either axial or terminal. Now what is this, sir? Axial or terminal? Can you explain? Yes, definitely. I am gonna explain this. So axial is what? Axial is axis. Let's suppose that this is my axis. Okay, like this is a branch, a plant branch, a branch of a tree. Okay, my head is axis. I hope so that you are able to see my head. Very good. So this is what? This is my head, and on my head you have kept a flower, right? So this position will be axial position. So in the similar manner, I'll draw this here. Let's suppose this is a branch and here we have the flower. So this will be axial position. And now let's suppose that I'm talking about the, oh, I'm very sorry, just give me 1 second. I just explained opposite. So terminal was this. Okay, if I'm here, standing here, the flower is here. So this position will be terminal. And what about the axial? So axial is basically what? Let's suppose this is the main branch and this is my hand, okay, and the flower is present here between the main branch and the other branch, okay. So this is what? This is the axial, okay, clear? Now I hope that it's clear. Now let's have pod or fruit color. Earlier we discussed what? Earlier we discussed about the pod or fruit shape. Now we are discussing about the pod or fruit color. So it can be what? It can be green and yellow. It can be green and yellow. And then we have the seed shape, the actual shape of the peas. It can be round and it can be wrinkled, right? Clear? But sir, you have given this thing, it's fine, we will be writing it, but it's very, very hard for us to learn. Yes, I understand that it may be hard for you to learn. So that's why I am here and I am going to help you with a very easy peasy trick. So let me give you a trick. Here, few things you have to keep in mind. So let's suppose there is a girl, okay, which is fat. Okay, the girl is what? The girl is fat. Imagine this, okay? The girl is fat and the name of the girl is Ria. And I'm very sorry for all the Rias watching the video. So the girl name is what? The girl name is Ria and she is fat. And where she is going? She is going to her village. So a girl, Ria, fat, going to village. And in village, what she is doing? She is yawning. She is yawning. So a girl going to village and yawning. What type of girl? A fat girl. So let's write here fat Ria. F-A-T. Fat Ria. Go to village. Fat Ria. Go to village and yawn. Fat Ria go to village and what they do? What she do? Yawn. Okay. Now sir, how we are going to learn this? Very easy. Fat Ria go to village and yawn. So F for what? F for full. F for what we can write here is that, see, F, if I'm talking about the F, so we can say here full, like flower position we can say here. Now A is what? A is for axial, okay. T is for what? Round, which is tall, okay. One second, what I have written? Just a second, just a second. Okay, so this one is fat Ria go to village, right? So for F, what we can write here? F we can write here full. A we can write here axial, right? T for tall. R for round. G for green. Again T we will be ignoring this, and then V for violet and Y for yellow, right? So we have what? 1, 2, 3, 4, 5, 6, 7 characters. We have this 7 characters, and what? How you can learn is that? You can learn it very easily. See here. We have this yellow, okay. Yellow will be dominant. Why? For what? Yellow will be dominant for which thing? See, the seed color, right? And you know that the seed color will be yellow dominant, okay. And then we have what? We have the violet, we have this green, we have this round, axial, and the full position, okay. Clear to everyone? Any doubts anywhere? Just let me know. Do you have any doubts anywhere? Just let me know. Okay. Now what is this for full? Okay. This full is basically what? This full shape. We discussed the inflated or constricted, right? So in place of inflated, we are just using the term full. The pod shape is what? The pod shape is full, inflated. Clear to everyone? I hope so you don't have any doubts. Now, let's move on. Let's discuss about the Mendelian cross. Okay, like what did Mendel do? How was the experiment? How he did the experiment? What were the results? Everything we are going to discuss. So we can discuss monohybrid cross today, and if the time will be there, we will be discussing dihybrid cross as well. If not, dihybrid cross we will be discussing tomorrow. Okay, now let's see. Let's go with the monohybrid cross. So before going to the actual cross, I would like to introduce some of the things to you. Let me find a blank page. Okay. I hope that you have written this: Fat Ria go to village and yawn. If you have not written it, after the class you can pause this video and make a note of it. Okay. So as of now, I am deleting this thing. Okay. Because why? I need a blank page. Next, see here, if I'm talking about this thing, so let's suppose that I have this capital T, I have this small t, and I have this capital T and small t. Okay, just I'm explaining the terms so that you will be able to understand the cross better. So this is what? This is a gene, right? A gene controlling the plant height. This capital T is a gene controlling plant height. This one capital T is what? This one capital T is a allele, is an allele. Okay, this is what? This is allele. And again, this capital T, single capital T, again this is what? This is another allele. So since this particular thing, which is the pea plant, is what? Is a diploid organism. So it will have what? It will have two pairs, right? It will have one pair, that means two alleles. So you can see one allele is this and one allele is this. And since the allele are same, so can I say that this condition represents the homozygous condition? This condition represents the homozygous condition. Yes or no? This represents the homozygous condition. And what about this small t? So this small t, again this is a gene which is controlling the plant height. This is a gene which is controlling the plant height. But here we have a different allele, but both the allele are same. So we are going to write here, again this is what? This is also homozygous condition. And in this condition, the plant will be short, while in this condition, the plant will be tall. Okay. So here, as we saw, that capital T is basically what? It is dominant. Yes. Capital T is what? Capital T is the dominant one, and small t is what? Small t is the recessive one. So in presence of recessive characters, in presence of recessive allele, which one will show its phenotype? Obviously the capital T, right? So in this condition, what's happening? Both the alleles are different, and this condition is called as heterozygous condition. Yes, this condition is known as heterozygous condition, and here the character, whether the plant will be tall or short. So I just said that if there is a presence of dominant allele, then recessive is not going to show its character. So yes, here we have the presence of dominant allele, which is capital T, and so this particular thing will have what? Will have a tall height. Okay, tall height. The plant will be tall. Clear? This thing is clear, right? Now let's suppose that, just for an example, I will be explaining some more things to you. So here, if I am talking about other things, like we have, just one second, I'll clear the screen for you. Okay, now let's suppose that we have one generation, mother, father. Now, this mother and father did coitus and they produced a baby, right? So this baby will be called as what? This will be called as their F1 generation. Okay, we will call this as the F1 generation. Clear to everyone? We will call this as F1 generation. Now, this baby and this baby will not reproduce, this baby will grow up and then it will reproduce. Okay, so let's suppose that this is gonna participate in reproduction with another baby. Okay, with another baby or another adult human being, and then they are gonna produce some again baby. Okay, so this is what, this is their second generation, right? In a simple manner, let's suppose like your grandparents, okay, then your parents, and then you. So I can say that your parents were F1 and you are F2. Clear? This is what you have to understand, what you have to remember. Now, let's move on with the P. So we discussed that P is what? P is a diploid plant, right? Diploid plant means what? Having two sets of chromosomes, right? So we can write that P was what? P plant is diploid. Yes, we all know that P is now diploid. But if I ask you how many chromosomes P was having, we know about humans, we know that human is having 46 chromosomes. Why I say 43, I don't know. So human is having what? Human is having 46 chromosomes. In a similar manner, P is having what? P is having in total 14 chromosomes. In total 14 chromosomes, right? P is having 14 chromosomes. Now, if I talk about this P, then what will be the haploid number of chromosome here? So obviously, how we can calculate 2n is equal to 14 and then n is equal to 14 divided by 2. Although I'm not a maths teacher, but I know this basic calculation. So n will be equal to what? 7. So 7 is what? 7 is your haploid content of the P, right? Now, here we have one thing. Here we need to understand one thing is that these 7 pairs of contrasting characters were present on this 7 chromosomes only, right? Yes, obviously, if we have 7 pairs of chromosomes, then all those 7 characters has to be present in these 7 chromosomes only. But is it necessary that all the characters will be present in one character in one chromosome, second in second, third in third, fourth in fourth, fifth in fifth, sixth in sixth, seventh in seventh? Is this how they are present in the chromosomes? No. So there were some chromosomes in which there were no characters present, which was chosen by the Mendel. The characters which was chosen by Mendel, some of the characters were not present in some of the chromosomes, and in some chromosomes it was what? It was maybe 2 or 3. Okay, so we are going to see that in which chromosomes what characters were present which was chosen by Mendel. So let's see here. Here we have what? And this was observed by a particular scientist named as S. Dixit. S. Dixit, okay? Now see. This was founded by the S. Dixit, and this person said that there were what? There were 1, 2, 3, 4, 5, 6 and 7 chromosomes. Okay, we know that 7 chromosomes are there, and this person said that on the first chromosome we had flower color, and on the first chromosome only we had seed color. So see, on the first chromosome we had 2 characters, right? And on the second chromosomes, no character. Like other characters would be present, but the characters chosen by Mendel was not present. Now let's go on the third chromosome, and in third chromosome also there were no characters. No. Then we go to fourth one, and in the fourth one we have plant height. Then we have what? We have pod shape, okay, and then we have flower position. Okay, these all were present in the fourth chromosome. Then let's go to the fifth one, and in fifth one we just have one character which was pod color. Sixth one was khali, empty, and then we had the seventh one which is seed shape. Okay, so these were the distribution of the chromosomes. Sorry, these were the distribution of the characters present on the different chromosomes. So if I say that which chromosome is having maximum characters, like maximum Mendelian characters, so chromosome number 4, you can see 3 characters in a single chromosome, right? And then if I say, what should I say? What should I ask? Should I ask which chromosomes doesn't have any characters chosen by the Mendel? So the chromosomes were 2, 3 and 6. So this 2, 3 and 6th chromosomes do not have any characters chosen by Mendel. Okay, so in exams they can confuse you by giving such statements that first, second, third chromosome number contains the Mendelian characters, and if you have not read this, you will be like, okay, let's do the inky pinky ponky, and whatever numbers you will feel okay, then you are gonna tick it and then come to exam, and then you are going to come home and you will see that congratulations, you have got a negative marking. So if you want to avoid this thing, learn this. 2, 3, 6, no chromosome. Just learn this. 2, 3, 6, no chromosome. Maximum chromosome number of, 2, 3, 6, no characters. Maximum characters on 4th chromosomes, right? Clear? Now, let's move on. Let's move on with the monohybrid cross. So, here, mono means what? Mono means single, sir. Not that single, okay? Single. Mono means single. So, in monohybrid cross, what we are going to discuss? Like how a single character gets transferred to the other generation, right? Again, how a single character, say plant height, a single character gets transferred to the second generation. A single character, let's suppose flower color, gets transferred into the second generation. If we are just focusing on the single character, then that is called as monohybrid cross. Okay, so now let's discuss the monohybrid cross in very detail. Dihybrid cross is what? Dihybrid cross, 2 characters. If I am taking 2 characters simultaneously, if I am studying that, okay, how plant height and plant flower color is being transferred to the next generation. Is there any relationship between them? Do they depend on each other? If this thing I am studying, then I'll go with the dihybrid cross. Clear, everyone? Okay. Now let's move on and let us talk about the monohybrid cross. Okay, so monohybrid cross is basically what? You can refer to the line which I have written here, which is cross performed to study the inheritance of a single character or a pair of traits. What is this? This is a very good line. This is a very good thing. It said that cross performed to study an inheritance of single character. That's very good. We totally understood, sir. But what is this pair of traits? See, a character is controlled by a pair of traits. Let's suppose that if you are talking about the plant height, so since this plant, which is Pisum sativum, is diploid, and since it is diploid, then what is gonna happen? It is going to have 2 alleles, right? Capital T, small t, or maybe the combination may be capital T, or maybe the combination can be capital T, small t, right? So this combination is what? This combination will give a tall plant. So this is what? This is a character. But if I'm talking about the pair of traits, so the allele is, this allele is going to give what? This allele contains the property to give tallness, and this contains the property to give shortness, right? So this is how we can say study of inheritance of a single character or pairs of characters, pairs of traits. Is it clear? Because tall and short was what? Tall and short was the traits, and plant height was the character, which in this case the plant height is tall, which can be said as character here, and this too is the traits. Clear? So this is what this line means. It's a very good thing, right? Very beautifully written, a single character or a pair of traits. Now let's move on and let's finish it, the monohybrid cross, quickly. So in monohybrid cross, the very basic example what we have got is that we have chosen a plant, not we, basically Mendel has chosen this. Mendel chose a plant, a character which was plant height, okay, which was plant height. Plant height. Clear? Now, if I am talking about this plant height, so how many traits we have? Sir, 2. One is tall and other is short. Very good. Now, I told you that Mendel used pure breeding or homozygous parents, right? Pure breeding or homozygous present. How was that obtained? Anyone in the comment section? How did we obtain pure or homozygous condition? We obtained that by the help of self-pollination, right? So we have here, if we are talking about the pure one, so we will have capital T and capital T alleles. Here what we are going to have, small t and small t alleles, right? Now they are going to reproduce, they are going to form the gametes, and we know that in gametes, if the body is diploid, the gametes will be haploid. Yes or no? That's great. So from here, let's take a single T. Let's take a single T. And from here also, let's take a single T. This is the gametes. These are the gametes, right? These were the gametes which were produced by whom? The plants, the Pisum sativum. And now we did the fusion of these gametes, and then we got what? We got capital T and small t as the progeny. So this can be called as what? F1 or F2? Fast, come on. This is called as F1 or F2. This thing is called as F1 generation. Is it all clear to everyone? This is how we obtained F1. Okay, I'm going to write it in single page. Just let me find, yes, we have got this. So what we did, we took homozygous pure parent, right? So let's write here homozygous pure parent. We have homozygous pure parent. Very good. We have this. And then what we have? We have the capital T, which is tall parent, and short parent. Okay. This tall we have written capital T, capital T. Why we have written? Because Mendel said that he is taking homozygous parent, right? Pure parent. And now we have made the gametes. So gametes will be what? Capital T and small t, right? These are the gametes. And then we are going to fuse them. After fusion, we got capital T, small t. And this is what? This is the F1 generation. This is what? This is F1 generation. Is it clear to everyone? Yes? That's great. Now, if I would like to ask you, how many plants are tall in F1 generation? How many plants? If suppose that you have taken 500 plants, okay, just for an example, you have taken 500 plants. Among 500 plants, how much you think that they are tall? 400, 200, 300, right? How much? All tall. What? All tall. Can you see any other combination? Can you form any other combination in this cross? You have got capital T and small t as the gamete. This plant is not going to give any other gametes. It is only and only going to give capital T. This plant is also only and only going to give you the small t. So they are not going to form any other kind of gametes, right? And that's why there is no other combination possible in this thing. So only and only we are going to get a tall plant, all tall, right? So how can I write the thing here? Like all tall. Yes, that means phenotype will be what? phenotype will be tall. Phenotype will be tall. Clear? Sir, what is phenotype? External characters we discussed in the last class. And then I'm going to discuss about the genotype. So here the genotype is, I'm going to have a question for you, like whether this genotype is homozygous or heterozygous. Whether this genotype is homozygous or heterozygous. Fast, I need the answer. Whether this genotype is homozygous or heterozygous. So this genotype is heterozygous. This genotype is heterozygous condition. Why, sir? Because one is capital T and the other one is small t. Two different kinds of allele is present. So can I say that pure parents produced impure progeny? Homozygous parent produced heterozygous progeny, right? So now my next question is that, let's have a question. All progeny in F1 generation. I want to check how many of you answer this question correctly. And if you are watching this video in recorded format, pause this video, comment in the comment tab, and I'm gonna check it. All progeny in F1 generation is phenotypically and genotypically similar to their parents. Now, here I'll give you the options, although I can't give you the poll on YouTube. The poll can be given in your special class, but still, you have the comment tab, right? Is this statement true or false? Now, tell me, is this statement true or false? How many of you go with true? How many of you go with false? Sir, it's very much confusing, right? See, this statement is completely false. This statement is completely false. Why? Phenotypically it's same. Is it? No. Phenotypically with only one parent it's same, right? So we have a tall plant and we have one tall parent, right? We have a tall plant in F1 generation and we have a one tall parent. So we can say that the phenotype of this particular progeny matches the phenotype of one parent. What about genotypes? Genotypes doesn't match with any of the parent. This particular kid is having capital T, small t as its genotype, and their parents are having capital T and small t as their genotype. So genotype is completely different, and phenotype also, it doesn't match with the both parents, right? Neither there was a blending. There was no blending also, right? No blending in this case. What does this blending mean, sir? Let's suppose that you took a white color, you took a gray color, black color. You mix it and you get a color which is between black and white, which is gray. But here, in this particular thing, it is not happening like you are taking the tall plant, you are taking the short plant, and tada, you will have a medium plant. No, this is not possible here. So I can say that there is no blending. So Mendel proved that. See here, see, there is no blending in this kind of thing. Mendel proved this, that blending doesn't take place. Okay, and with the help of this cross, he gave some laws, postulates, which we are going to discuss. Now, we got this F1 generation as capital T and small t. Now, sir, what we have to do? With this F1 generation? We are going to do the self-crossing. We are going to do the self-pollination. So here we have capital T, small t. And since we are going to have the self-pollination, the other parent will also have what? Capital T and small t only. Clear? Now let's form the gamete. Now if we are going to form the gamete, so what gametes we are going to get? From here we are going to get capital T, from here we are going to get small t. In the similar manner, from here also I am changing the color. If I have taken white for the first one, let's take white for the first one only, right? And where is my orange color? Yes, we have this. So we have this capital T and small t. So how many types of gametes do we have? How many types of gametes do we have? In total, how many gametes do we have? 4 gametes we have, right? Okay. Now, we can do something like this. Tuck, 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 something like this. And we can have the answer, but that's not a good method. So we are going to follow the NCERT, and NCERT says to make a proper box and then arrange the gametes and then make the progeny, right? So that box is known as Punnett square. Let's make Punnett square. Someone says Punnett square also. So it's your wish, whatever you need to say, you can say. Now let's have this Punnett square. See, how many gametes we have? 4 gametes. So we have to accommodate this 4 gametes. So let's suppose this was the male plant and this was the female plant. So male plant will write here. It's not necessary, you can write male anywhere, female anywhere, okay? So let's write male here, so capital T, small t, and let's write our orange female here. Okay, now let's make the progeny. How we are going to make? This capital T will fuse and form capital T, capital T. This capital T and this small t will form capital T, small t. Similarly, this capital T and this small t, sir, small t, yes, small t. This small t and this capital T will make capital T and small t. In the similar manner, this small t and this small t will produce small t, small t. Clear? Okay, we got it. Now let's write this thing again. So we have capital T, capital T, capital T, small t, capital T, small t, small t, small t. This is what we have got from this particular cross, where it is gone. Yeah, this is what we have got from this particular cross. I hope that you are able to see this cross, right? Yes, here what we have, capital T, and below that what we have, capital T, small t, right? Then what we have, capital T, small t, and then we have small t and small t. Okay, now let me come back to the camera. So let's discuss this thing. What will be the phenotype of this particular genotype? Obviously this will be tall. Again, this will be fast, and whoever stays short is not going to crack the NEET. So say it properly. Is it what? Is it tall? Yes, sir, it's tall. Is this one tall? Yes, it's tall. What about the cap, small t, fast? What about the small t, small t? Small t will form short plant, right? It will form short plant. Clear to everyone? Yes, please see this. If then I ask you to form or found a ratio among the small short plant and the tall plant, then how you're going to found it? Sir, all these, if I write here tall and if I write here short, I know it's not visible to you, I'll just be little side. So this is what, this is tall and this is short. So how many tall, sir? 3 talls. How many tall? 3 talls. How many short, sir? 1 short, right? So 1 short. Yes, we are going to have 1 short sessions also in this channel. So what will be our phenotypic ratio? What will be our phenotypic ratio? Oh, did I said that this is F2 generation? No? Okay. Now I'll say this whole thing is what? This whole thing is F2 generation. F2 generation. Now, here we have phenotypic ratio. Come on, fast. Phenotypic ratio is what? Phenotypic ratio is, where is my pen? Yeah, 3:1. Phenotypic ratio is 3:1. That means among 4 plants, 3 plants were tall and 1 plant was short. Among 4 plants, 3 plants are tall and 1 plant is short, right? Clear? Now, what is the genotypic ratio? Genotypic ratio. So to find out this, let's first of all see that which one are same and which one are different. Okay, so this is same. Yes, this both are same, capital T, small t, capital T, small t, right? And this are different. So the ratio for this one will be 1, the ratio for this one will be 1, and the ratio for this one will be 2. If suppose that we have 4 plants, okay, if suppose that we have 4 plants, and in 4 plants, 2 of them are similar. So what will be the ratio here? 2, okay, it will be 2. What will be the ratio of this? 1 and 1, right? Clear? So how we can write that the ratio of genotypic ratio will be 1 is to 2 is to 1. The genotypic ratio will be 1 is to 2 is to 1. Is it clear? 1 we are having capital T, capital T. Second we are having capital T, small t, and the third we are having small t and small t. Clear? The first one is going to produce tall plant, the second one is going to produce tall plant, but the third one is going to produce the short plant. Now why this second one is able to produce tall plant, sir? Because it is having capital T, the dominant allele, and if it is having a dominant allele, it is going to produce what? It is going to produce the tall plant. It is going to produce the tall plant. Okay, clear? Now before ending this session, let's have a beautiful question, a very easy peasy question I'm going to give, okay, and you have to solve this question. So are you ready for it? If you are not, then also I'll give you the question and you have to solve this. So let's have a question that the question says in a Mendelian cross about 1600 plants, about 1600 plants were in F1 generation. Okay, F1 generation of monohybrid cross. We are talking about the monohybrid cross, and my question is how many have dominant phenotype. So this question is what? This question is asking that Mendelian cross, okay, and in Mendelian cross in F1 generation, in F1 generation of monohybrid cross, how many of are dominant phenotype? Okay, so option 1, let's have 1200, option B, let's have 1600, option C, let's have 1000, and option D, this may be the answer, right? 500. Okay, giving you a minute, pause this video, solve this question. And many of you are gonna get trapped into my trick. Okay, the one who was saying 500 is the answer, it's wrong. It's not just like because I focused on that 500 and okay, that has focused, this might be the answer. No, it's wrong. Now we are talking about the F1 generation, right? If I'm talking about the F1 generation, all plants were tall. All plants were tall, so 1600 will be the answer. Clear? If in the same question I ask you for the F2 generation, same question, same thing, just replace F1 with F2, okay? Or I'll do it. I'm replacing this F1 with F2. Then what will be your answer? Come on. Let's have the answers in the comment section now. Okay, how many answers of how many plants will have dominant phenotype? So the answer to this thing is what? The answer to this thing is 1200. Okay, the answer to this thing is 1200. How, sir? How, sir? See, dominant, we had this, okay, we had 3:1 ratio of phenotype in F2 generation. 3 plants were tall, and one plant was short. So in the similar manner, if you are taking 4 plants, in 4 plants, 3 is tall and 1 is short. So in 1600 plants, what is going to happen? In 1600 plants, 3 by 4th plant, 3 by 4th of 1600 plant will be what? Will be tall. So by solving this, we are going to get 1200, right? How, sir? This also I am not gonna do. You know basic maths, right? But still, I will do. 4 ones are 4, 4 twos are 8, 4 threes are 12, 4 fours are 16. So 4 fours are 16, then we have 2 zeros here. Put it 3 fours are 12, then we are going to get 1200 as our answer. So 1200 plants among 1600. Among 1600, how many plants? How many plants are having dominant phenotypes, sir? 1200 plants are having dominant phenotype. And how many plant is having recessive phenotype, which is short? So 1600 minus 1200, if we will do, then we will got what? We will get 400. So around 400 plant is having short phenotype. Okay, and let's check the work of Mendel. Was he true or not? So if we divide 1200 upon 400, we are going to get what? 3:1 ratio. So in F2 generation, whatever the number, we will get 3:1 ratio. And this is how you're going to solve this type of questions. In the next class, which will be tomorrow at 6:30 p.m. sharp, we will be discussing about the dihybrid cross. And in the next to next class, we will be solving all those important questions which can be asked in NEET from these particular 2 chapters. Till then, bye-bye everyone. Do share this session among your friends and like and comment if you have watched this lecture. Then you are definitely gonna crack your NEET. And if in case you have any doubts, I will be available in the comment section. So you comment, I'll comment, and let's clarify our doubt. Till then, bye-bye everyone. See you in the next class.